Hello everyone, I hope you are doing well. Welcome to my channel Biochemistry Club, and today I am here with the third lecture from my lecture series on glycolysis. So this lecture is about fates of pyruvate, and my major focus will be on the process of fermentation. So let's start today's lecture. As I have discussed in my first lecture that after its entry inside the cell, the glucose managed to produce two molecules of pyruvate by the process of glycolysis. And once the pyruvate is produced, next the further fates of pyruvates will depend upon oxygen availability. So if there are aerobic conditions, if oxygen supply is sufficient, then the pyruvate will move from cytosol to mitochondrial matrix. And in mitochondria, there is an enzyme PDH complex, which will convert the pyruvate into the acetyl coenzyme A. So this process will take place inside the mitochondrial matrix. And once the pyruvate has been converted to acetyl coenzyme A, this acetyl coenzyme A will be entered into the TCA cycle and further oxidation will take place. During TCA cycle, the NADH and FADH2 will be produced, and these reduced electron carriers will finally be used in the third stage electron transport chain for the production of ATP. So this is how the complete oxidation of glucose will takes place. The process of glycolysis doesn't depend upon oxygen availability. The presence or absence of oxygen does not affect this process. But the further two stages, the TCA cycle and electron transport chain, these two processes will take place only in aerobic conditions. So if you are engaged in high muscular activity, in high physical activity, or if there are hypoxic or anaerobic conditions, then glycolysis will take place, but the pyruvate will not enter into the TCA cycle. So this fate will stop in anaerobic conditions and pyruvate will be converted into either lactate or ethanol. So there is a specific reason for this fate, for the conversion of pyruvate to lactate or for the conversion of pyruvate into the ethanol. And these processes are sometimes called as fermentation. So what is the reason behind fermentation? Why fermentation is necessary in anaerobic conditions? In order to address this question, in order to answer this question, I will revise a concept which I have previously described in one of my lectures, that is about NAD plus regeneration. So please keep one point in your minds clear, that the amount of NAD plus, the oxidized form or the reduced form of electron carriers in different compartments of the cell is limited. So suppose in cytosol there is a limited quantity of oxidized form of NAD plus is available. And during the process of glycolysis, when glucose is being converted into the pyruvate, there is an enzyme, the first enzyme of the payoff phase, which actually converts the glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This enzyme requires NAD plus. So by the activity of this enzyme, one molecule of NAD plus is converted into the NADH. So actually what is happening when the glycolysis is taking place, the oxidized form of NADH is being converted into the reduced form by the action of enzyme glyceraldehyde 3-phosphate dehydrogenase. So a moment will come when all oxidized form of NAD plus has been depleted and it has been converted into its reduced form. So what will happen if the glycolysis wants to take place? We must regenerate the oxidized form of this electron carrier. We must have to reconvert this reduced electron carrier into its oxidized form. So for this reduced electron carrier must have to donate its electron to some other species, to some other molecule. So where these reduced electron carriers, they will fuel their electrons, where they will release their electrons. Definitely they will release their electrons to any molecule whose reduction potential is greater than their own reduction potential. And inside the cell, there is an organelle, mitochondria. The inner membrane of mitochondria contains some enzymes or complexes which belongs to the electron transport chain. And the final electron acceptor of this chain is oxygen. So what will happen during aerobic conditions? These reduced electron carriers, they will indirectly enter to the mitochondria through some shuttle systems like malate aspartate shuttle. And inside the mitochondria, they will release their electrons and they will donate their electrons to the complexes of electron transport chain. So what will happen once these reduced electron carriers, they will give their electron to the complexes of electron transport chain. They will be oxidized and oxidized electron carriers will move out again indirectly and NAD plus will be regenerated by this means. It means electron transport chain is the only way to regenerate NAD plus during aerobic conditions. So complex 1 of electron transport chain will transfer or donate this electron to the complex 2, which will transfer its electron to complex 3, and these carriers will finally donate or transfer the electron to molecular oxygen, which will be then reduced to the water. So by this means, the NAD plus is going to be reoxidized or regenerate. But there is an important question. If the conditions are anaerobic, if the conditions are anaerobic and if oxygen is not available, then what will happen? So in anaerobic conditions, the final electron acceptor in electron transport chain is not available. Complex number 1, complex number 2, complex number 3, and complex number 4, but oxygen is not available. So what will happen when NADH, they will release their electrons. The electrons will be transferred from complex 1 to complex 2 to complex 2 to complex 3 and 4, and there is no other species or no other acceptor available to pick this electron. So these complexes, they will refuse to accept further electrons from NADH. It means this normal route, which was previously available for the reoxidation of NAD+, will not be available in anaerobic conditions because oxygen is not available. Oxygen is mandatory for the reoxidation of NAD+. So if oxygen is not available, electron transport chain is unable to reoxidize the NAD+. So what is the other pathway? What is the other route available for the reoxidation of NAD+? Or how these NADH, they will manage to reoxidize themselves, because we all know that if these reduced electron carriers, they will not be reoxidized, the process of fuel oxidation, the process of glycolysis will stop. So in anaerobic conditions, because aerobic respiration is not taking place, the complete glucose oxidation is not possible. So the process of glycolysis must have to take place. We must have to reoxidize the NAD plus. So how this NAD plus will reoxidize? Some of the pyruvate which is produced by the process of glycolysis, it will be converted into the lactate or ethanol. It means pyruvate in anaerobic conditions will not move to the mitochondrial matrix. It will not be converted into acetyl coenzyme A, but it will stay inside the cytosol, where by the action of some enzyme like lactate dehydrogenase or by alcohol dehydrogenase, it will be reduced to the lactate. And during this process, what will be happen? NADH will transfer its reducing equivalents to pyruvate and it will be reoxidized. So by this means NAD plus will be reoxidized and the process of glycolysis it will remains continue. So this is what we call the process of fermentation during which the pyruvate is managed to during which the pyruvate will be converted into the lactate or ethanol. Why? Only to regenerate NAD plus. So let's see how the lactic acid fermentation takes place. So when animal tissues cannot be supplied with sufficient oxygen to support aerobic oxidation, the pyruvate will be reduced to the lactate and during this process NAD plus will be reoxidized. As you can see in this reaction, that lactate dehydrogenase enzyme will come forward and it will transfer the reducing equivalents from NADH to carbonyl carbon of pyruvate and will reduce this carbonyl carbon to lactate. And during this process, the NAD plus will be reoxidized, which will be then used in the process of glycolysis. So in order to make the process of glycolysis continue, in order in order to make it sure that the process of glycolysis will be continued, the fermentation will take place. Similarly, the ethanol fermentation is similar, but it is unique to some microorganisms, mostly yeast. Here, the enzyme alcohol dehydrogenase will convert the acetaldehyde to ethanol. So here we need two enzymes. First, the pyruvate will be decarboxylated by the action of enzyme pyruvate decarboxylase. And then the two carbon fragment, the acetaldehyde, it will be further converted into the ethanol. So decarboxylation was necessary to convert the pyruvate into ethanol. And the enzyme alcohol dehydrogenase, it will further be used to reduce this carbonyl carbon. And during this oxidation reduction reaction, the reducing equivalents from NADH will be transferred to the carbonyl carbon. And by this means, the acetaldehyde will be reduced to ethanol and NADH will be reoxidized. So fermentation is actually a type of strategy by which the cells they manage to reoxidize NAD plus. The sole purpose of fermentation is to reoxidize NAD plus during anaerobic conditions. But keep this point in your mind that the lactate produced during the process of fermentation is not a waste, because once the condition will become favorable, this lactate will move towards liver, where the process of gluconeogenesis will convert this lactate back into the glucose. So this is a type of management strategy where the cells during the period of anaerobic conditions, they convert pyruvate to lactate, but once the conditions are restored or replenished, the lactate will again be converted into the glucose. So there is no wastage of energy because this glucose will again be reoxidized and it will produce the same ATPs which it can produce in normal aerobic circumstances. So the only difference in the process of fermentation and glycolysis is that there is no NADH production in the process of fermentation, because the two NADH that will be produced during the process of glycolysis, these two NADH will be reoxidized when the pyruvate will be converted into the lactate. So we can say that during the process of fermentation, there is no net gain of NADH. The glycolysis will convert to NAD plus to NADH, and the process of fermentation will reoxidize. It will reconvert NADH to NAD plus. So there is no net gain or no net production of NADH in fermentation. Only the glucose will be converted into the two lactate or two molecules of ethanol along with the production of two ATPs. Okay, now the last point is the regulation of glycolysis. And as I have discussed in my first lecture, that the major regulatory enzyme of glycolysis is PFK1. But in addition to the allosteric regulation, there are hormonal regulation as well. And enzyme hexokinase, PFK1, and pyruvate kinase, these are the other enzymes which are subjected to the allosteric regulation. So when there are low energy molecules available, when AMP and ADP, they are present in high amount, high concentration, this is an indication that cell is in the state of starvation, and these molecules will act as activator for all these enzymes as a modulator. Similarly, the high amount of ATP is an indication that cell is in well-fed state and there is no need of ATP. So the process of glycolysis will halt in these conditions. Similarly, the hormones glucagon, epinephrine, and insulin, they can also affect the process of glycolysis. Okay, now there is a last point which I want to mention here, and this point is about the effect of insulin on glucose uptake. We all know the cells, most of the cells of the body which have glucose dependent transporters, GLUT4, they cannot uptake the glucose in the absence of insulin. So, what is the normal pathway? Suppose this is the glucose which is present in blood. After a meal, when we are in well-fed state, there is a gland that is pancreas, and this pancreas, its cells, they keenly observe the concentration of glucose in the blood. If there is high blood glucose concentration, the pancreas will release a secretion that is insulin. And if there is low concentration of glucose in the blood, the alpha cells of pancreas, they will release another secretion that is glucagon. So the pancreas is a gland that is responsible for releasing certain secretions in blood in response to glucose concentration. So in well-fed state, when there is high glucose concentration, it will release the hormone insulin. And this insulin will help the cells to pick up the glucose from the blood. And why some cells cannot pick up glucose in the absence of insulin? This is because the normal transporters of glucose, the glucose transporters, they are present inside the cytosol along with a vesicle. Because the gates of glucose or the transporters are not available in plasma membrane. On the other hand, the insulin receptor is always present on plasma membrane. So what will happen when the concentration of glucose is high, pancreas will release insulin and insulin will come and bind to its receptor and it will convey a message to the cell that glucose is available in plenty. So the transporters will come and they will attach to the plasma membrane and glucose will now then enter inside the cell. So if insulin is not available, the message will not be conveyed and glucose will not be entered. You can see this through this diagram that insulin will first bind to its receptor And then this vesicle will fuse to the plasma membrane. It will insert its transporter, glucose transporter to the plasma membrane, and then glucose will come inside the cell. So majority of the cells which are glucose dependent, majority of the cells which are insulin dependent, they have GLUT4 types of the transporters which are normally sequestered inside the cell, and they will come into the plasma membrane only in response to insulin. So this is the reason if insulin is not available, the most of the cells of the body, they cannot pick up the glucose. So I hope this lecture has been helpful for you. If you have any question, you can ask in comment section and I will try to answer those questions. Thank you.